Hello. We know that DNA or the genome of the bacteria is double-stranded and it is circular. So if replication did happen in them, they would have happened in such a way that the initiation would have happened. So we'll just have it this way, and then a transcription bubble and formation of bidirectional replication fork like this, and the replication forks would travel onto the other side. And eventually what we will get is two DNA molecules, and they are interlocked in something like this. And then DNA topoisomerase would have separated them into two independent or separate DNA molecules, which would then be segregated to the daughter cells. So that is pretty straightforward and simple in a way. But when it comes to eukaryotes, there are numerous chromosomes per genome set, and they are linear. And because they are linear, there is a problem, what we refer to it as N replication problem. Say this is a linear DNA molecule, and we have one replication fork here, and this is another replication fork. It moves on, and usually eukaryotic chromosomes do have multiple origins of replication. And as the replication is happening, if we can notice that at the last, when it comes to around this stage, it would have to have synthesized a small primer, and then the DNA polymerase, that is DNA polymerase epsilon, would have synthesized, and this RNA primer would have been deleted by DNA polymerase 1, say for example. If it were a eukaryotic RNase like enzyme, the homologous one would have removed the RNA primer. But now this gap cannot be filled by anything. If it is possible, it could only be filled up by RNA polymerase. But that's not what we want. We already just now removed the RNA primer that was already there. We need it to be DNA. And this is a problem because there could be some essential genes. And then if this chromosome undergoes a round of replication, further round of replication, the DNA will become shorter and shorter after each round of replication. And that means the cell is likely to lose many more genes as they are that are in the periphery, and gradually it will not produce a complete genome set, because of which the daughter cells will be deficient or they will be non-productive. Here is a nice resource, that is Khan Academy. I hope you already know it. It's free, and there are a lot of things to brush up our basics, and it's a fantastic resource for learning from children to even adults. I am Dr. BCMR, Assistant Professor at Shastra. Today we will discuss about the N replication problem and how nature solves it. As we have already seen, what N replication problem means is the N replication problem is the trouble with these missing links here and here. And we need to have a way of getting rid of or filling those gaps. Especially assume I'm going to put, say there are some genes like this A, B, C, D, E, F, G and H. In this case we have H is missing, I mean it's not double stranded here, and in this case A is missing. And if we could not fill these gaps, they will be vulnerable to be lost. And if replication happened, then the genomes that are produced would have lost both the H completely on both the strands. So there should be a problem, there should be a need to fill these gaps. And nature has different strategies to solve these. And here, what could be the strategies? If we think of them, then we can see it as some of the strategies is the problem. First of all, it comes because DNA polymerase does not have, it is absolutely or obligately dependent on 3' OH. It cannot de novo start polymerization by itself. RNA polymerase can do that. RNA polymerase is a little bit special, the functions of which are different than that of DNA polymerase. DNA replication happens once per cell cycle, and it only has to start in one place, and because it has to be highly coordinated with that of this cell reproduction, it cannot happen just randomly anywhere. So only certain designated places that we call as origins of replication is where DNA polymerase starts initiation, and that too, it is dependent on RNA polymerase. RNA polymerase, on the other hand, all these genes that are indicated here should be transcribed in different cells to different extent, and then it doesn't need to be, or shouldn't be dependent on something else to initiate. Of course, it should be regulated which genes should be expressed and which ones should not be expressed, for which transcription factors and many other factors are involved. But the point is, DNA replication and RNA polymerization, or DNA polymerization and RNA polymerization happen, they have different purposes. DNA replication is to maintain or multiply so that the progeny could get a complete set of the genome. So it's highly regulated based on cell division. RNA polymerase or RNA polymerization is dependent on how cell responds to the surroundings, and the response is in the form of differential gene expression. Different genes are expressed, and therefore RNA polymerase has it, and DNA polymerase lost the ability to initiate replication by itself, evolutionarily speaking. At this point, what we know is DNA polymerase does not have the capacity to initiate replication by itself. If it had, it could have filled this gap very easily, but that's not what is happening, isn't it? So how else can we depend? Now, the problem or the strategies, if we look at, so all DNA polymerase requires is a OH here, based on which it should be able to fill these gaps here like this. There is another strategy. This strategy would be to, if I could extend this longer, and then there is a possibility for primase to come and fill this gap, and then this DNA polymerase could take up and fill it this way, and later we can have more and more, and later these RNA primers could be removed and filled up with DNA. So the H gene, which is on the borders, is surviving. The same thing will happen on both the sides. That is a possibility, and this is how exactly nature solved these problems. And there are many other different types of solutions nature has come up, but we will only discuss two of them. The first one is based on some viruses. They have special DNA polymerase, and they have a protein called as priming protein. Priming protein, which has a OH group from one of its side, probably it's a threonine residue that gives the OH that is present here, and the OH can be used by some of these DNA polymerases, special ones of course, and then they will start filling up the gaps this way, and eventually we will have this whole DNA strand synthesized, and from here you will get this. So there is no more end replication problem to start with, because they are using a special protein. It is called as priming protein. There is another other strategy that is quite popular in all of the eukaryotes, if you see, and the viruses are actually good in that sense to study, because their solutions, or the solutions we find in viruses, seem much more diverse and enthralling actually. The one popular in all of our cells is what we call it as telomerase. The strategy of extending the strand this is the overhang, and this gap needs to be filled. So we have 5 prime here, assume that, and this is 3 prime. So we need to fill this gap. So the one problem point is to extend this template longer. If we have a long template, then the bottom strand can be synthesized, so the bordering genes can be saved or rescued in that sense. So what happens here is there is an enzyme called as telomerase. Telomerase has 2 components, protein component and the protein component shown in the green, and the RNA component. Here is the sequence and 5 prime, and the sequence and the 3 prime. So the RNA component is used as a template here for extending of the top strand in this case. So what it does now is to fill up these gaps. So it will fill up complementary G, G, T, T, A, G. And the next step is translocation. What it means is this the telomerase, which is an RNA polymerase, which is a RNA dependent DNA polymerase. If you remember about reverse transcriptase, it is one of the examples of a reverse transcriptase. Like some of them, some of the viruses do have specialized transcriptases. This is for the purpose of extending the telomeres. So what happens in this case is the telomerase now will move and translocate to a different location, something like this. Right? Previously it was somewhere here. We filled up this gap was filled up by us, by the telomerase, and now once that small region is polymerized, telomerase would move this way, and that step is called as translocation. And now again polymerization occurs, and the same way it keeps on moving, and it extends for a large into a large space, and it will add multiple of these polymerization steps, because of which the DNA of the top strand here is extended long. Okay, this way it keeps going on. So once it has reached several hundred of it, RNA primer may come, the primase may come, and then it would synthesize a small primer. Assume this is a primer, and then because this is the 5 prime and here is 3 prime, now the bottom strand or the complementary strand will be synthesized this way. What we need to appreciate is the ingenuity of how the evolution of such an enzyme happened, and how it keeps maintaining the telomere size. So quickly, telomerase is a RNA dependent DNA polymerase. It's something similar to that of reverse transcriptase. It has an RNA component, and it has the RNA component does act as a template for the extension of the overhang, and based on that the other one is made. If we look at the structure of the RNA template, you can see a certain kind of symmetry, and that symmetry is important or essential for the steps to happen. You can see that this is a region AAU, and it is followed by another same repeat of AAU, and that is what allows the telomerase to move from one location to the translocation step. It can only translocate if it has complementary strand, right? Something like that. So I hope you understood, and one will be able to realize the importance of the strands here, or the symmetry of the template here. AAU followed by another AAU, and that symmetry is important. Say for example, if I have given you a sequence or 4 sequences and try to say which one of it is possibly the sequence or in the RNA template of a telomerase, what would you give? This is 1, 2, C, and AGG. So out of these, the first one is not an option because I mentioned that it is RNA, and RNA doesn't have thymine. So that's a quick win. Although it follows the symmetry, but the base is wrong, so it's not RNA. The second one is not because it doesn't follow the symmetry that we discussed. The third one does. Obviously, I have to give one correct option. And what does happen at the tips of the telomeres is the question. There are several proteins that protect the ends of the telomeres, because at the end one overhang will be left eventually. So there are proteins called as RAP1, RIF2, RIF1. They coat the telomeres, they bind to them, and the ends are coated by CDC13, something like this. And the DNA itself folds upon itself, because the ends, something like this, is vulnerable to attack by exonucleases. Therefore, it loops and base pairs upon itself like this, so that no ends are freely available to the exonucleases. Therefore, the chromosomes are much more stable, without which they would have been digested away. The important functions of telomeres there are three important functions. First is to solve the end replication problem. That is, by extending, they are solving, or they're reducing the number of the losses of the genes that are present in the borders or the edges of the chromosomes. Second one is they would prevent the degradation. By forming these chromosomes and the T loop, the ends are not accessible to the exonucleases, thereby they are giving structural or informational stability to the chromosomes. The third one is also important. Say, for example, you have a chromosome and this is another chromosome. There is a possibility for these two to recombine, and because telomeres are present here, the recombination is prevented, and that's how the chromosomes will remain as independent chromosomes rather than fusing together. So that is also another important prevention of recombination between chromosomes. I mean, here we are talking about something like non-homologous recombination. Yes, and what we need to also understand in this context is that telomeres have telomeres, if we see the expression, they are highly expressed in stem cells, even embryonic stem cells mostly, and they are also interestingly expressed highly in cancer cells. The cancer cells, they do divide continuously. That is the prominent feature of cancer cells. That means as the stem cells, when they divide and they could keep on differentiating, they lose the activity, telomerase activity, and eventually if they keep on dividing, the differentiated cells, they would not have as much telomerase activity, therefore the chromosome shorten, and then the cells age and the cells will die. But cancer cells, they would keep on dividing. So one way of targeting cancer cells is to target the telomerase. If we target the telomerase, probably they would not be able to divide or produce productive cells, thereby killing the cancer cells. I hope I have given you some ideas about how nature solves problems, especially in the cases of the telomeres end replication problem and telomeres and their significance. Thank you very much. See you in another video.